Hello friends, welcome to my channel. I'm in coaching center. I'm going to explain your that is the adult and biology chapter 11, biotechnology principles and process processes. Let's come to the chapter. Let us know about the 10 recombinant proteins which are used in medical practices. Let us know. Recombination proteins are obtained from the recombinant DNA technology. These are insulin that is obtained by the treatment of type 1 uh, diabetes mellitus and interferon used for the chronic hepatitis. Interferon is a DA that is used for herpes and viral enteritis, and coagulation factor that is 7 treatment for hemophilia A, and coagulation factor 11, the coagulation factor 9 that is treatment of hemophilia B, and DNA 7 treatment of cystine, and antithrombin 3 that is prevent of blood clot, infectant B for treatment of multiscleral and human recombination growth hormone for providing growth in an individual and tissue, plasminogen activator treatment of actual myocardial infection. Next, let us know about the chart. The sites of which the PMA or DNA site. Here, see, this is the DNA sites here, and the product it produces here. This DNA sites like this we have marked here. Next. Hmm. Next, let us know about the bigger or DNA is bigger in molecular size. Enzymes are smaller in size than DNA. This is because DNA contains genetic information from the development of functional of all organisms. It contains the instruction of synthesis of proteins and DNA molecules. On the other hand, enzymes are proteins which are synthesized from a small stretch of DNA known as the genes. Total number of chromosomes into 6.023 into 10 to the power 23. Therefore, 46 into 6.023 into 10 to the power 23. 2.77 into 10 to the power 18 molecules. This is the diploid cells in human. Next, let us know about the eukaryote cells have restriction. No, eukaryote cells do not have restriction endonuclease. This is because DNA of eukaryote is highly methylated by a modified enzyme called methylase. Next, let us know about the better aeration and mixed properties and what are the advantages. The shake flask method is used for a small scale production of biotechnological products in a laboratory. On the other hand, stirred tank bioreactors are used for large scale production, and the advantages of small volume of culture can be taken out from the reactor for sampling or testing. It has a foam breaker of regulating of foam, and it has a control system that regulates the temperature and pH. Let us take the 5 examples of palindromic DNA sequence. Let us take and let us palindromic sequences is a certain sequence of the DNA that reads the same whether and from 5 to 3 direction or from 3 to 5 direction. They are the sites for the action of restriction enzymes. Like this it will be, like this. Next, can we recall about the meiosis and indicate what stage is recombination DNA is made? Meiosis is a process of involvement that involves the reduction of the amount of genetic material, and it is two types, namely meiosis 1 and meiosis 2. During the Pachytene stage of phase 1, crossing over of chromosomes takes place where the exchange of segments between non-sister chromatids of homologous chromosomes takes place, and this results in the formation of recombination DNA. Let us know about the next is the how a reporter enzymes can be used in the monitor transformation of host cell by foreign DNA. A reporter gene can be used to monitor the transformation of host cell by foreign DNA. They act as a selected marker to determine whether the host cell has taken up the foreign DNA or the foreign DNA taken up the or the foreign genes gets expressed in the cells. Next, let us know about the origin of reflection and bioreactor and downstream processing. So, origin of replication is defined as the DNA sequence in a genome from where replication initiates, and bioreactor are large vessels used for large-scale production of biotechnology products from raw material. And downstream processing is a method of separation and purification of foreign products after a completion. Next, let us know about the PCR, restriction enzymes and DNA and chitin. So polymer PCR is a polymerase chain reaction is a technique in molecular biology to amplify a gene or a piece of DNA to obtain its several copies. Hmm. Restriction. Hmm. And restriction enzymes are molecular scissors used in the molecular biology for cutting DNA sequences from the special sites, and they are of two types, that is exonuclease or endoclase. Exonuclease plays an important, it is a type of restriction enzyme that removes the nucleotide from 5 to 3 5 or 3 end of DNA molecule, and endonuclease, it is a type of restriction enzyme that makes the cut within a DNA at a specific site. Next, chitaceans are class of enzymes used for the degradation of chitin, which forms a major component of the fungal cell wall. Next, let us know about the difference between plasma DNA chromosomal DNA. So plasma DNA is an extra chromosomal DNA molecule in bacteria that is capable of replicating or independent of chromosomal DNA, and chromosomal DNA is an entire DNA of an organism. Next, RNA. RNA is a single celled strained molecule and it contains ribose sugar, and the pyrimidions in RNA are adenine and uracil, and RNA cannot replicate itself and it is a component of ribosomes, and DNA is a double-stranded molecule and it contains deoxyribose sugar, and the pyramid in DNA are adenine and thymine, and DNA molecules have ability to replicate and it is a component of chromosomes. Let us know about the exonuclease and endonuclease. So it is a types of restriction that now we learned here. So it is a type of restriction enzyme that removes the nucleotide from 5 or 3 ends of the DNA molecules, and it is a endonuclease. It is a type of restriction enzyme that makes a cut within a DNA at a specific size of generate sticky ends. So okay friends, this was your chapter. So this was your chapter biotechnology. So like the video, comment on my subscribe my channel.